Hey everyone, welcome to Professor Long's Lectures in Microbiology. I'm Professor Bob Long. As you know, these videos are intended for use by students who are enrolled in my courses at Belmar College. Anyone else who uses these videos or finds them useful, please hit like or leave me a note, subscribe to the channel, and I'll get a good idea of whether I should keep doing this or not. Anyway, these videos are done pretty crudely, pretty quickly, in a short period of time. It's a cell phone, me, and the board. We're doing this mainly due to the COVID shutdown and the quick delivery that I need to get this information to students. Now, by no means is this an exhaustive overview or exhaustive detailed analysis of all the information that's available to you guys. These are intended to be fundamental and foundational principles that are presented so that you guys can add more detail later. This is intended for health science majors. It's a microbiology class for the health sciences. Now, in a previous video, chapter 14, the video before this one, we were covering chapter 14 in our textbook, which is sort of the transmission of microbes, disease and epidemiology, and then how microbes are and pathogens are transmitted. What I originally started to do was combine chapter 14 and chapter 15 of our textbook, and I actually presented some of the chapter 15 material at the same time. And in this video, as we cover chapter 15, which is the pathogenicity and pathogenesis of microbes, we're going to repeat some of the same information. It's okay if we repeat it. The more often you hear something, the better you learn it and understand it. Anyway, so let's get started. When we talk about pathogenicity, what we're talking about is the likelihood that you would get ill from a particular pathogen. So pathogenicity refers to the capability of a pathogen of producing disease. What is the likelihood that you could get sick from something? Virulence refers to the intensity of the disease or illness that you experience. How sick is it going to make you? So pathogenesis is, will I get sick or not? What's the likelihood I can pick this up? Virulence is, how sick will I be once I have it? So pathogenesis is the capacity of a pathogen to generate a disease, and virulence is the intensity of the illness produced from that pathogen. Now, one of the things that I think we covered previously was sort of the factors, the factors, sorry, it's getting cold out and my respiratory tract is dry, so my throat is dry. So some of the factors that can predispose a person to an illness, and we've covered this I believe before, but I want to run through some of these factors just briefly. One of them is a person's gender. And again, we live in a time where some people are trying to move towards a genderless society. But when it comes to certain medical conditions, we have to know what the birth gender of a person is. And you have to be identified as having either being male or female. And one of the reasons is that some illnesses are much more likely to affect one gender over the other. For example, the human papillomavirus can cause genital warts in males and females, but in females it can cause cervical cancers. So since males don't have a cervix, they're not susceptible to the cancer-causing nature of papillomavirus. Also, certain disorders affect males more. For example, meningitis is one of the disorders or illnesses due to a pathogen that tends to infect males more than females. And another one that affects females more than males would be urinary tract infections due to fecal bacteria, simply because of the anatomy of the human body. So gender is one of the major issues. Inherited traits or your genetic makeup. So some human beings may lack a particular gene or enzyme that makes them more susceptible to some disorders than others, or may have some kind of genetic variation. One of the best examples is sickle cell disease. So for individuals who have sickle cell disease and the sickle cell trait, they have a gene that causes the red blood cells to alter their shape. One of the benefits of that is they have a much lower likelihood of contracting malaria. We know that climate and weather can affect things. So some colds and some upper respiratory tract infections are much more prevalent in colder climates and colder times of the year. And a lot of tropical diseases are much more prevalent in warmer regions and warmer parts of the world. So the climate, the weather, or the geographic location may make you a little bit more susceptible. Age affects how likely we might be to pick up certain illnesses or be affected by certain pathogens. As you know, when we're really young and we haven't developed immunity because we haven't had a lot of exposure to diseases, then some of the young are pretty susceptible to many of the diseases and disorders. When we get older and our nutritional habits change, our diet changes, our body starts to break down, our energy levels start to decline, then you can be much more susceptible to certain illnesses and diseases. Age can have an impact sometimes on how susceptible you are to a specific pathogen or illness. We talked about fatigue, being tired. When we exhaust all of our energy, your immune system that fights infectious agents and pathogens requires a tremendous amount of energy. First of all, the white blood cells will divide rapidly by mitosis and increase in numbers when we're infected with something as they encounter a pathogen. That alone takes a tremendous amount of energy, as does all the protein synthesis and the formation of antibodies and some things we're going to talk about in the next lecture or two. So simply being exhausted means your immune system is not going to have the energy available to divide and fight certain infections, and we become much more susceptible. Lifestyle is another issue. So people who are IV drug users, people who have a lot of sexual partners and unprotected sex or very promiscuous sex, very often can be much more susceptible to sexually transmitted diseases or bloodborne diseases, or simply transmitting a disease from one person to another. A lot of IV drug users will pick up hepatitis from each other from sharing needles and things, as well as AIDS, HIV, some other diseases. Nutrition has a huge impact. So a healthy diet with good vitamins and a very well-rounded diet is really important for giving your body the energy and repairing damage to membranes and skin and things, so that we can ward off some of these pathogens. And finally, we can talk about immunocompromised individuals, individuals whose immune system has been suppressed. If someone's had an organ transplant and they're on immunosuppressant drugs, then their immune system is being kept at bay, which would make them susceptible to invasive pathogens. Also, people who are on things like chemotherapy, a lot of chemotherapeutic drugs for cancer treatment will decrease the or suppress the immune system, making you much more susceptible to certain diseases and pathogens. Another thing I want to talk about is what we call the 7 challenges for any pathogen to enter the human body and to cause illness. There's what we call the 7 challenges of pathogenesis for an organism. And these challenges include these things. And then we're going to go into some detail into some of these. One of them is the ability to maintain a reservoir. So what I'm going to do is I'm going to write these out first. Let me make sure I'm writing within the scope of the camera. Yeah, I am, but I'm going to move this over just a little bit. Okay? I want to leave some room so that we can expand upon these. So the ability of a pathogen to maintain a reservoir. And again, let me get all these down first, and then we'll talk about each one in more detail. We'll simply go down the list. But essentially what a reservoir is, where a pathogen will live or reside between infections. So when it's not infecting someone, sometimes a pathogen can live in an organism, and we call that organism a reservoir. Again, we'll go into a little bit more detail in just a minute. Then it has to have the ability to leave the reservoir to enter a host. Okay? And the host for us is the human body. So it has to have the ability to maintain a reservoir, a place where it will live when it's not causing the infection, or between infections. It has to have the ability to leave that reservoir. There's a portal of exit or a way that it can exit, and to enter a host. Okay? Now, in order to enter the host, several steps have to occur. One is it needs to adhere to the host or the host cells. So it has to have a way to stick to the cells. So essentially, we're going to have some kind of animal that has some infectious agent in it. Maybe it's the rabies living in a possum or something. And then the infectious agent has to have a way to leave that organism. Might be through a bite, it might be through some other way. And that reservoir does not have to be an animal reservoir. Again, we'll go through this in more detail. But then it has to actually adhere to the cells of the host. This would be the nucleus of a host cell, and these pathogens have to have a way to adhere to the host cell. Okay? So they have to maintain a reservoir, leave the reservoir, adhere to a host cell. And then they have to actually be able to invade the host. So not only just stick to it, because we have some things that can stick to our skin that don't make us sick, but they need to be able to invade the host and enter the cells and or the tissues where they would reside. And so in this instance, we would actually have some pathogen entering our cells somehow, some way. A virus injecting its genome, or our cells engulfing and phagocytosing or endocytosing some foreign pathogen. Now, once it's invaded the host, it needs to have the ability to evade the host defenses. Our immune system, really. It has to find a way to get away from our white blood cells. So very often, some of our cells, once infected, can express proteins on their surface, and white blood cells can come gobble those cells up and kill them. Or we might have some bacterium that has entered the body, and our white blood cells can engulf it, phagocytose it, fuse that with the lysosome and digest it, and kill the pathogen. But what if a pathogen has something like a capsule around it, a slime layer that prevents it from being engulfed? So they have to have some way of evading the host defense system or the immune system so that they can cause an infection. Now usually when we take in a pathogen, you don't take in a ton of them. So they need the ability to multiply within the host. They need to increase in number. And sometimes the amount of a pathogen that enters our body is not enough to make you sick. And so there's a thing called the infectious dose and the ID50, which we'll go over in a minute. And in order to cause illness, there's a certain number of bacteria that must, or a pathogen that must enter your body. Or if a low number does, they can multiply inside the body and then cause the illness. Okay? And then finally, the last step is they have to have the ability to exit and return to a reservoir. So, these are 7 challenges that any pathogen must overcome in order to cause an illness and continue to be pathogenic. They have to maintain a reservoir, somewhere that they stay between infections or can be found and live. They have to leave the reservoir to enter the host. They have to adhere to the host or the host cells. They have to invade the host cells. They have to evade the immune system so they don't get wiped out before they cause illness. They need to multiply or have a high number in order to cause the illness. And then they need to be able to exit and return to the reservoir to continue to propagate and infect other individuals. Okay? So now, I'm going to leave these on the board, and I want to leave some room here for us to go and break down each one of these. So when we talk about maintain a reservoir, basically a reservoir is a place where the pathogen can maintain itself when it's not infecting the host. So it's a place where they live when they're not causing infections. Now, reservoirs can be numerous. They can be human reservoirs. So someone who's ill is considered a reservoir. If you have the flu and you walk in a room full of people, you're a reservoir that can then transmit the disease to others if it's a communicable disease. Now, healthy people, people who are not exhibiting signs or symptoms of the illness, can also be a reservoir. So you don't have to be ill, you can be healthy and not sick from the disorder, and we call those people carriers. So if you're a carrier of a disease, you're considered a reservoir. And there's different types of carriers. One type is called an incubatory carrier. An incubatory carrier is someone who's carrying the disease while it's still kind of incubating. They have not begun to experience full-blown signs and symptoms of the disorder or disease. They're just having mild symptoms and not really feeling the effects of it. So if, let's say, someone contracted the flu and you just start to notice a little bit like, man, I don't feel 100% right now. I don't feel right. I might be coming down with something. You would be called an incubatory carrier. If you are someone who has the disease, but it doesn't show signs or symptoms for a long period of time, those individuals are called a chronic carrier. So when we talk about a reservoir, when we talk about humans, someone who is ill or healthy, called a carrier. And then if we talk about an incubatory carrier, this is prior to full onset of symptoms, or we could say signs and symptoms. Let me make sure I'm not writing off the board here. If I am, I need to, no, I'm just on the edge, so I'm going to readjust my camera just a hair so that I have a little bit more room to write, okay? So, if you're considered a chronic carrier, chronic meaning long-term, then a chronic carrier is someone who's carrying the disease and harbors the pathogen over an extended period of time. So it's someone who harbors the pathogen for an extended period, for a long time, essentially. So, for example, someone who has HIV for several years but isn't expressing the disease would be considered a chronic carrier. Okay. Now, other types of reservoirs. So I'm going to erase some of this information because I will run out of board. There are animal carriers. Now, when we talk about animal carriers, different animals can carry diseases. For example, a lot of rodents, bats, possums, a raccoon, a cat, a dog, anything that carries rabies would be considered an animal reservoir or an animal carrier. And so it's any animal that can harbor a pathogen would be called an animal reservoir. Now, if there's a pathogenic disease that humans can contract from an animal, we call that a zoonosis. You can think of a zoo. So a zoonosis is any pathogen causing illness in humans that is carried by an animal reservoir. And And a good example of that would be rabies. So, well, there's some others we could talk about, but I'm not going to spend too much time on this. Now, finally, there are some organisms that can transmit disease from a reservoir to humans, and we would call that a vector. So I'm going to erase this, and we're going to talk a little bit about the term vector. So a vector is anything that transmits a disease to the host. Okay? And in this instance, the host is going to be a human being. So we can have vectors that transmit the disease, and sometimes those vectors are living organisms, like arthropods, mosquitoes, ticks, lice. They can transmit a disease from, say, a deer or another animal to humans. They're not necessary to the reservoir. They're the vector that transmits the disease. The reservoir was in the animal. So you don't have to be bitten by an animal reservoir like in rabies. If a rabid dog bites you, you could pick up rabies. You could have something like deer tick disease that is carried by deer, and then if a tick bites the deer and then gets on you while you're out in the brush and bites you, then the tick would serve as the vector. The animal would be the reservoir, and it would still be considered as zoonosis because it's a pathogen carried by an animal that can cause disease in humans. Now, there's also environmental reservoirs. So we've talked about animal reservoirs, we've talked about human reservoirs, and then there are environmental reservoirs. Environmental reservoirs would be things like soil, contaminated water, or things that we call fomites, which are inanimate objects that can carry a disease. For example, a cup or a dirty bed sheet that isn't changed or something in a hospital. Certain clothing, that can carry a disease to some people. You could get athlete's foot from somebody else's shoe. If you wear their shoes, that would be considered a fomite, an inanimate object that can transmit a disease. Okay. Now, so those are the reservoirs that I wanted to mention. There are human reservoirs, there are animal reservoirs, there are environmental reservoirs. Now, the second key is that, or the second challenge is that the pathogen has to be able to leave the reservoir in order to enter or infect a host. And when they enter the host, we're going to talk about a thing called a portal of entry. So, portal comes from the Latin and Spanish word puerta, which means a doorway. So this is how they enter the body. The portal of entry is essentially the part of the body where the pathogen enters, which is kind of self-explanatory when you look at the name. So, where do pathogens enter the human body? We have external portals and we have internal portals. And so the external portals would be the outside of the body. The two main culprits for external portals would be the skin and the conjunctiva or conjunctiva of the eye. A lot of bacteria can live in our skin, and they can get in through sebaceous glands and sweat glands. That's one way that they can enter. As a matter of fact, it's a very common way that a lot of pathogens enter the body through our skin, is actually more so through sweat glands and sebaceous glands than, say, openings and cuts. And then the conjunctiva of the eye. If someone sneezes and you get some droplets with some flu virus or something on your eyeball, or something that's transmitted by respiratory droplets, then you might be able to absorb that through the body or into the body. Internal surfaces would be things like mucous membranes. So we have the oral cavity, the respiratory cavity, digestive tract, the urinary and reproductive tracts. All of these are lined with mucous membranes. Essentially, if you can stick a finger in it and it's nice and moist, then it's a mucous membrane. Through your mouth, through the nose, most of the respiratory tract going down into the lungs, through the digestive tract as we ingest things, the urinary and reproductive tract are portals of entry inside the body that many pathogens can enter, okay? So, now when we talk about the internal and external surfaces, one of the things I do want to mention, I'm going to erase this, is what we call parenteral. Okay, parenteral or parenteral. When we have parenteral transmission, this is when a pathogen enters through a break in the skin. Things like a bite, a cut, or wound. Okay? If a tick bites you and injects, or a mosquito bites you and injects the pathogen through the skin into underlying tissues, then that would be called parenteral transmission. And so they can enter the skin sometimes through natural openings like the sebaceous glands and sweat glands, or we can have parenteral transmission where we have a bite from a rabid dog or an insect or an arthropod. You have a cut on your skin that some bacteria get in, or some kind of wound on the body that becomes infected. That would be called parenteral transmission. Now, two concepts I want to mention when we talk about this is I want to talk about the ID50 and the LD50. So, when we talk about the ID50, it tells you infectious dose 50%, okay? This is defined as the infectious dose, okay? Now, when we look at the ID50, one of the things that we're talking about is we're talking about what dosage does it cause of what number of the bacterium or pathogen entering the body is required to cause an infection. So, this is really defined as the number of microbes that must enter the body to cause an infection. So, infectious dose, the dose or number of microbes, like a dose of medicine, so to speak, that must enter the body that cause an infection. Okay, I should say, and 50% of the, I'll say host, but really very often what you do is you're actually looking at in test animals, okay? The host is usually what we would call a test animal. We don't want to test this on humans, although some people are very upset that things are tested on animals as well, but it would be considered to test unethical and immoral to test things on humans to cause an infection in a human who is otherwise uninfected. So, the infectious dose, 50, is the number of microbes, the dose that it takes, to cause an infection in 50% of the test animals. Now, there's another concept, and I'm just going to change the color of this, called the LD50, which is the lethal dose, okay? Or the lethal dose 50. Now, the lethal dose 50 is the number of microbes that must enter the body to cause death in 50% of the test animals. That's called the lethal dose 50 or LD50. The number of microbes that must enter the body in order to cause death in 50% of the test animals. Okay, so we can look at the ID50, the number that are necessarily to cause an infection in 50% of the animals, or the lethal dose 50, the number of microbes necessary to cause death of 50% of the host animals. And so whenever a pathogen does enter through one of the portals of entry, then we can look at what is the LD50 or the ID50. How many have to enter? If only a small number get in, then our immune system might wipe them out before they cause infection or could lead to death. So we need to know these numbers. Now, when we have communicable diseases, those are spread from one person to another. There's a number of ways that communicable diseases can be spread from person to person. One can be through aerosolized droplets, like when we cough or sneeze or we're talking, we're hacking things out or laughing out loud. One can be through direct body contact, so that would be like kissing someone or having sex with someone where you're exchanging body fluids. There's what we call vertical transmission, and this is one I want to focus on a little bit, and I've talked about it in the last video. But in vertical transmission, this is when the disease is carried from parent to offspring, from the mother to the child, so to speak. Where in horizontal transmission amongst humans, then we communicate the disease or transmit it from person to person that are not the parent-child relationship. Like I said, like making out with someone, having sex with someone, or coughing and sneezing near people. But in vertical transmission, we're going from one generation to the next, or from the mother to child. And there's two ways that this can happen. One is called prenatal transmission. This is during pregnancy or prior to birth. Nate, and from nature, and naissance means birth in French. So this is during pregnancy. The pathogen can cross the placenta somehow, some way, sometimes through the umbilical cord. But we call that prenatal transmission. If we use the term perinatal, and do not confuse this with parenteral. Peri means around birth. In perinatal transmission, this occurs during birth. Okay? If a mother has some kind of bacterium in the vagina, in the birth canal, an STD, sometimes they can be transmitted to the offspring. There's even some that can cause blindness in children. So you need to know that in vertical transmission, prenatal means during pregnancy, it crosses the placenta. Perinatal is one that's picked up by the offspring as it passes through the birth canal or during birth. Okay? Now, those are types of communicable diseases that are transmitted from human to human. Another way they can be done is what's called the fecal-oral route. So if someone goes to the restroom and doesn't wash their hands very well, or wash their hands at all, and they go eat, or they touch a doorknob and someone else touches it, and then scratches, picks their teeth or something, or licks their fingers while they're doing something, then you can transmit from fecal to oral. So it goes from feces through the oral cavity simply by touching something that's been contaminated with fecal bacteria. There's one that we don't, that's really not on the notes, but there's also the UTIs. Many urinary tract infections are caused by fecal bacteria that make it into the urinary tract. Because on males, the way that the genitalia is so far, the opening for the urethra, the urinary tract is so far from the anus, it's very difficult for males to get urinary tract infections. But females who do not practice proper hygiene, particularly if you have like a baby or an adult that is in diapers that needs to be changed, and they defecate and then they don't get changed for a while, then some of the fecal bacteria can enter into the vagina and into the urinary tract and cause urinary tract infections. Also, wiping the wrong way is another way. or certain types of underwear are much more likely to, thongs, for example, can transfer bacteria if they slide around from the fecal, from the anus to the vagina, and end up causing urinary tract infections. Now, when we talk about airborne transmission, we're talking about things that can be passed through droplets in the air and survive quite a while. And then vehicle transmission, when we talk about vehicle transmission, I'm not talking about the transmission in your car. Vehicle transmission would be through fomites, inanimate objects that can carry things like, a cup that's got bacteria growing on it from someone else drinking out of it. So drinking after someone, using someone else's clothing that hasn't been washed, or beds and sheets and pillows and pillowcases that aren't properly cleaned or sterilized. Surgical tools that haven't been properly sterilized, those would be considered fomites. And then there's what we call the parental transmission, where we take an insect bite. We've talked about vectors and all of that. So that is how things can leave their reservoir and can be transmitted through vehicles, through vectors, through fomites, through aerosolized from human to human contact. Now, when it comes to our third challenge, these pathogens have to have the ability to adhere to a host or to host cells, to stick to them. And so some pathogens have on their surface molecules called adhesins. Adhesins are proteins that allow a pathogen, and only certain pathogens have them, to adhere or bind to host cells. So in some instances, a host cell might have a protein on its membrane that allows this microbe to adhere to it and stick to the cell. So we call these adhesins, simply proteins that can bind to specific receptors on host cells. Now, many of these adhesins can be components, molecules that are found on the capsule of some bacteria, or also found associated with things like pili of bacteria. So very often they can be on the surface of the cells or on the surface of the pilus. So some bacteria have an outer covering called the capsule, and very often these adhesins can be a part of that, or if they have a pilus, some bacteria have pili on them that have adhesins on those pili that allow them to adhere to host cells. Okay? Now, let me see if I want to go into any more detail on this. I don't think I do. We can talk about a few other things, but that's really one of the main topics. I don't want to go into too much biochemistry of this stuff, the type of bonds that are formed. But once they adhere to the cell, then the invader has to be able to invade the cell. The pathogen has to be able to make it inside the cell or inside the tissue. And once they penetrate deep into body tissues, we call this an invasive pathogen. Invasive pathogens can invade deep into body tissues. So if you have some kind of invasive pathogen, they have the ability to pass into the body and invade tissues that are deeper inside the body. Some of them actually live inside the host cells. So if you have a pathogen that actually makes it inside the cell somehow and lives inside the cell, then we call those intracellular parasites or intracellular pathogens. All obligate intracellular parasites are intracellular pathogens. So intracellular simply means inside the cell. One of the things we know is that rickettsias that cause Rocky Mountain spotted fever and chlamydias can very often live inside the host cells. Now, once they've invaded the cell, and you know, some pathogens actually make it inside the cell because they get engulfed or enclosed inside of an endocytic vesicle, and they get endocytosed, and then they can live inside the cell that way, or at least enter the cell and cause their infection. Now, once they're inside the body of the organism or inside the cell, they have to be able to evade the defense mechanisms. They have to be able to invade the immune system. And this is a big deal. One of the things that happens is some bacteria have a capsule. And not only can the capsule have certain proteins like the adhesins that we talked about a minute ago, but some capsules can make a pathogen slippery, so to speak. This is sort of a way that people describe it. But basically, in some instances, the capsule can block phagocytosis by immune cells. So it makes them kind of slippery where they sneak away because the immune cells cannot recognize them or cannot bind to them. And so the capsule of some bacteria plays a role not only in allowing it to adhere to host cells, but also allowing it to evade the host cell mechanisms. Some cells have what we call surface proteins called M proteins. M proteins also prevent phagocytosis of the bacteria unless the bacterium is labeled with antibodies, and there are antibodies that can bind to some of the M proteins. But essentially these M proteins can serve like little hair-like projections on the bacteria. Now these are different than flagella, but they're like little hair-like projections. Maybe they're not that long, but what happens is that it doesn't prevent a white blood cell, or it prevents a white blood cell from being able to phagocytose these bacteria. So some bacteria have a capsule that prevents phagocytosis, some have these M proteins. And then some again can invade inside the white blood cells. Once they're inside the white blood cell, then the white blood cell doesn't have the ability, or the antibodies don't have the ability to get in them. For example, HIV replicates inside of white blood cells. So does Mycobacterium tuberculosis, by the way, that causes TB. Now, another way so they're on the external surface of many bacteria, they have proteins. They either have a capsule that allows them to evade phagocytosis, or the M proteins on the surface. Another way that they can evade our immune system is, well, I really don't want to go into all the enzymes in great detail, but what we can talk about is antigenic variation. Okay? I might go into the proteins or the enzymes, but they're called exoenzymes. Antigenic variation. What does this mean? Well, some bacteria have proteins in their outer covering, in their membrane, and they'll have different membrane proteins, lots of these, all over them. Our white blood cells, or we call these surface antigens because they can generate an antibody response. So some of our white blood cells, called lymphocytes, can release proteins called antibodies that can bind to the surface antigens on some cells and sort of tag them so that other white blood cells can come in and wipe them out. Well, let's say your lymphocyte has certain antibodies, I should say, that have receptor sites on them in what we call the variable region, that allow them to bind to specific surface antigens. And since all of these similar bacteria will express that surface antigen, then anytime we have antibodies to one, we have antibodies to all of that particular type of bacterium or that particular pathogen. It could be a virus, with one of the capsid proteins. But let's say while these bacterium or virus or whatever organism is multiplying, then when they divide, either by binary fission or whatever mechanism that they spread, what if while they're replicating their genome for division or for propagation, that they have a mutation to the gene for that antigen? And so as this cell, before it gets caught and starts to infect someone or is actually caught by the immune system, what if while it's dividing, it has a mutation and now that antigen takes on a totally different shape because of some genetic mutation? Now, our antibody might catch this old one, but the new version of it would not be detected by our immune system. So we call that antigenic variation. A change, we vary the surface antigens so that the immune system can no longer recognize that mutant of the strain. This is how some viruses and some bacteria can avoid our immune system. There is a thing that we can say is the production of exoenzymes. Again, we're talking about how we evade host mechanisms. Some organisms produce what we call exoenzymes. Exo means out of or outside, and enzymes are enzymes. And essentially, some organisms can secrete enzymes out of their body or out of their cells that will prevent our immune system from recognizing them or allow them to hide for our immune system. One of them is called coagulase. I'll give you a couple of examples. Okay, ase means it's an enzyme, and to coagulate means to clot together. So coagulation is the process of blood clotting, and what coagulase does is it triggers the blood clotting mechanism. And if we were to form, say, we have an infectious site, we have a site where there's a whole bunch of bacteria, and we form a blood clot around it, very often that will prevent white blood cells from penetrating and engulfing the pathogen. Or they can also clot blood and prevent any white blood cells from leaking into the site. Essentially, it walls them off from our immune system. So coagulase is one of these exoenzymes that triggers coagulation or blood clotting that allows bacteria to protect themselves or certain pathogens to protect themselves from our immune system. Another one is called IgA protease. And so it's kind of hard to go into detail in some of these because we haven't talked about immunoglobulins. Ig refers to immunoglobulin, which is a type of antibody. Okay. And so what happens is when we study the structure of an antibody, which we're going to do in the future, antibodies have it's really made up of 4 proteins. There's 2 long chains called the heavy chains because they're bigger and heavier, and they're chains because they're chains of amino acids. And there's 2 light chains, and these things are stuck together by some chemical bonds. One part of the molecule is called the constant region because the amino acid sequence here is constant. It never changes. But these 2 little spots here are referred to as the variable regions because that's the part that makes one antibody different from the other is the amino acid sequence here. This part of the amino acid sequence is constant from any antibody made, but is the variable region that makes one antibody different from another. There happens to be one called immunoglobulin A that can bind to certain organisms. Well, IgA protease, a protease is an enzyme that digests a protein, and IgA protease digests the immunoglobulin A molecules so that they can't bind to certain pathogens. Okay, so that's another version of what we call an exoenzyme that allows them to evade our immune system. One would be coagulase, which causes coagulation or blood clotting to protect the bacteria, and IgA or immunoglobulin A protease, which dissolves the protein called immunoglobulin A. And we may talk about IgA and IgG and IgE and IgM and all these immunoglobulins in the future. Okay. There is one that I do like to talk about. There's one more exoenzyme called streptokinase. Streptokinase dissolves clots. It is an enzyme that is used to dissolve blood clots. And essentially, in some instances, you have some pathogen that is engulfed or trapped in a blood clot. Well, it might protect them from detection, but then they can't spread. Well, streptokinase will dissolve the blood clot and now allow the pathogen to spread. So that's another example of an exoenzyme. So before I finish up here, let me make sure we review really quick. The 7 challenges for pathogens to be able to cause an infection in a human or in a host cell: they have to be able to maintain a reservoir, a place that they can live when they're not causing an infection. They have to be able to leave the reservoir and be transmitted to the host, in this case, humans. They have to adhere to the human. We've talked about different types of reservoirs: animal reservoirs, human reservoirs, and environmental reservoirs. We've talked about some of the molecules that allow them to adhere to things called adhesins. They have to be able to invade the host and be able to go get inside, parental transmission and other things. They have to be able to evade the host defenses, okay? And we've talked about several things that allow them to do that. We talked about the M proteins, we've talked about exoenzymes, we've talked about the capsule. So you should know some of the ways that they are able to evade. An antigenic variation, which is an important one. They have to have the ability to multiply within the host. We're not going to go into detail on that. We've talked about how certain organisms multiply, whether it's through binary fission like in bacteria, or budding like in yeast, and some viruses have the lysogenic phase, or have the ability to do budding where they pop off little vesicles that contain the virus. And then they have to be able to exit and return to a reservoir, which they would do so through what's called a portal of exit. Just like there's a portal of entry, there's a portal of exit. And by the way, some organisms have the same portal of entry and same portal of exit. So if they're blood-borne diseases, the portal of entry and the portal of exit would be through blood, through an open wound. Some enter through the respiratory tract and exit through the respiratory tract. Okay. I guess one of the last things I want to talk about, and it's really important, is when we talk about the ability to multiply, we're not always talking about the ability of the pathogen itself to multiply, but also for the ability for some toxin, some toxic chemical that can cause the signs and symptoms of the illness needs to be released into the organism somehow. And I want to spend one last little part on this area, and this is really important. You should know this. But the 2 most common ways that some bacteria can cause pathogenesis is through toxins, and then simply by evading the body's defense mechanisms. But the toxins that they produce are what very often make us sick. And those toxins are chemicals that are produced by the pathogen themselves. 2 major classes of toxins that we can talk about are called endotoxins and exotoxins. Now, excuse me, I apologize for drinking so much during this video. It's probably not, I should step more off camera when I do that. I usually do. And as a matter of fact, I am going to step off camera because I have some hot tea here. And it's a lot more soothing for my throat than the water. Now, when we talk about toxins, I'm going to erase all of this because we've gone through these 7, and I didn't spend a lot of time on number 7 other than the portal of exit. But what I do want to talk about is one way that pathogenicity or pathogenesis occurs is through toxins. Toxins are compounds, chemicals, that cause the illness. So some bacteria make us sick because of the toxins that they release. And there's two major classes of toxins. There's exotoxins. These are secreted out of the bacterium. The bacterium makes them, so they make some toxins, and then they just simply secrete them and release them outside of themselves. And those toxins, those chemicals, are actually what trigger the illness, our immune response, and make us sick. And then there are what we call endotoxins. Exo means outside, so these are released out of the bacteria. Endotoxins means inside, and really they're not necessarily always inside, but bacteria have a cell membrane, and then they have a glycocalyx, the cell wall, and then some bacteria have outside of that a capsule. Okay? And that capsule can very often sometimes be called like a slime layer, or it's called a capsule depending on how thick it is. So endotoxins are simply made by living bacteria and then are released from the bacteria. Exotoxins are toxins that are released by bacteria when they die. These are produced by living bacteria and released. These are only released when the bacteria die. I thought I said that backwards for a second. One of the things we know about exotoxins is that they can be released by what we call gram-positive or gram-negative bacteria. So there are some gram-positive bacteria that can release exotoxins, and there are some gram-negative bacteria that can release exotoxins. And they're always secreted into the surrounding tissue where they do their damage. When it comes to endotoxins, these are by gram-negative bacteria only. Only gram-negative bacteria release some of these endotoxins. Now, when it comes to the exotoxins, some of the exotoxins are named for the specific type of tissue that is affected. So, this marker is very hard to erase once it writes on the board. But these affect specific tissues sometimes. So, for example, one class are called neurotoxins. Well, guess what? Neurotoxins affect the nervous system and neurons. Enterotoxins are in the gut. The enteric bacteria that cause these disorders and diseases of the bowels can release toxins that damage the gut lining or make us really sick, called enterotoxins. Okay, so sometimes the exotoxin is named for the tissue that it affects. Now, another thing that happens is exotoxins very often can enter our bloodstream and cause what we call toxemia. So anytime we have an emia, that's when something has entered the bloodstream. Emia comes from heme. So bacteremia means there's bacteria in the blood. Viremia means there's viruses in the blood. Toxemia means there's some kind of toxin in the blood, usually an exotoxin. And by the way, when we do consume these, they call an illness that's due to high presence of exotoxin a toxemia. So toxemia is simply, not a toxemia, an intoxication. Toxemia means the toxins in the blood, and intoxication means you've ingested the toxin somehow. A good example would be like the botulinum toxin that you take in. It's produced by the Clostridium botulinum that can grow in like canned foods, canned meats, and then you eat it and you ingest it, then you can get an intoxication. You take the toxin into your body. Another thing that happens is this. There's a thing called antitoxins. Now, all of this is for exotoxins right now. There are things called antitoxins. These are antibodies that bind exotoxins. We have antibodies that can bind to exotoxins, and we call those specific antibodies antitoxins. Like an antivenin is a particular antibody that can bind to a snake venom, for example. Now, this allows us, by the way, to create somewhat of a vaccine for certain toxins. If we could take the part of the toxin, the epitope that causes the illness, or at least triggers the immune system, and expose some immune cells to it, they'll develop antibodies so that when they are exposed to the full antitoxin, we can bind it up and not get sick. And one of the things that we do know is that some exotoxins, since exotoxins are usually proteins, they are almost always proteins released by gram-positive or gram-negative bacteria. Some of them can act as enzymes, and they can have a detrimental effect on the host. There's some that can lyse red blood cells called hemolysins. They kill red blood cells, or they actually, what the hemolysins really do is digest the heme molecule and prevent red blood cells from carrying oxygen to tissues, and therefore you can sort of die from almost like suffocation. You're not getting oxygen to tissues. The tissues become ischemic and anemic, don't have any oxygen flow, and the cells can die. And there's different types of hemolysins. There's a whole other class called leukocytins. So when I talk about these enzymes, the hemolysins are ones that lyse red blood cells, hemolysin, and leukocytins are some that kill white blood cells and prevent your immune system from breaking down or finding the exotoxin. Now, all of this is true of exotoxins. For endotoxins, since endotoxins are not proteins, they are actually molecules that we call an LPS, which stands for lipopolysaccharide. Okay? It means lipid with many sugars. Lipopolysaccharides, and they are part of the outer membrane of gram-negative bacteria. This is my abbreviation for bacteria, the beta sign. So, gram-negative bacteria have a lipopolysaccharide outer membrane that can cause people to become ill, because within that outer membrane there are these lipopolysaccharides that, when released, will make a person extremely ill. And so these are not secreted from living bacteria like exotoxins are exuded or released, secreted. This happens when these bacteria are killed, and then the molecules get released and can cause toxicity within humans. So they are part of the outer membrane of gram-negative bacteria. They're not proteins, they're lipopolysaccharides, and they are only released when we kill the bacteria, and they can float around in the body and make us sick. One of the things that we do know also is that we don't tend to produce antibodies against these, and so there are no antitoxins for the endotoxins, only for exotoxins. And finally, they're usually not very specific for a type of cell or tissue. They make you sick and can damage cells wherever they go. Anyway, I think that's everything that I want to talk about this for now, because we're about to go into the immune system, and we're going to go into innate immunity, or non-specific immunity, and acquired immunity or specific immunity. And we're going to go into great detail into those areas, and it's a lot of information. So I think I'll stop here. Anyway, I do hope you learned something. I hope this was helpful to some degree. I don't feel like these videos are as good as I want them to be, but I'm trying to produce these really quickly under duress. And I got a million things flowing through my mind as to what I want you to know, and trying to trim the material back. So anyway, forgive me if I'm a little choppy in these videos, but I hope that they're helpful. I hope that they provide you some guidance. If you're in my class, you can print the notes and then follow the video and highlight. If you're not in my class, well, at least hopefully these videos help you in some way. I hope you had as much fun as I did. I hope you learned something. I hope I see you in the next video. Thanks for watching.